Welcome everyone. I'm Sabi Rao, Director of Scientific Planning here at the McGovern Institute, and I'll be chairing the session today. Thank you all for being here. It's my very great honor and pleasure to introduce Robert Desimone, the current director of the McGovern Institute and the Doris and Don Berkey Professor in Neuroscience. So Bob has really conducted classic and foundational work on attention throughout his career, and he continues to do so. But he's also just a tireless leader of the research here at the McGovern Center, playing a leading role in the launch of, for example, the Hock Tan and Lisa Yang Center for Autism Research and the Poitras Center of Psychiatric Research. And I see every day the tireless work that he does throughout the Institute in helping, for example, push forward the generation of new model systems for examining psychiatric disorders. And without further ado, I'll turn things over to Bob. And it's fitting that his talk title, which is an overview of current research at the McGovern Institute, is Precision Neuroscience, a Journey Through the McGovern Institute.
Thanks, Sabi. I know many of us probably have an image of Pat McGovern in our minds. And I'll tell you, the image I have is not of Pat up there, always smiling down on us. But my image of Pat is really much closer. I would say, looking over my shoulder. And I imagine Pat really all the time looking and sort of judging, like, how are things going? And are you really making progress, in the trajectories that we had all agreed on? And I regard today as kind of a 5-year progress report to Pat. I'll be giving you an overview of things going on in the McGovern Institute, and then you're going to be hearing some speakers from the McGovern Institute here at MIT and our sister institutes in China, and we'll see what Pat thinks. Now, as you heard, Pat and Lori founded the McGovern Institute, and I would say our mission statement has kind of evolved in that, of course, it's as its base, the understanding of the brain and the mind. And that's always been the foundational work of the McGovern Institute. But I would say that we have evolved, and the board has formally endorsed, a second part of our mission, which is to take, when possible, that knowledge we develop about the brain and apply it in ways that will help people. And I think Pat was always really dedicated to this idea that ultimately neuroscience is going to help people, primarily through health and brain disorders, but you imagine that there would be a really widespread impact on society. And that's what we've been trying to do. Now, you heard Phil Sharp, the founding director of the McGovern Institute, give an absolutely marvelous account of the history and development of the Institute. And I would say our philosophies that we follow today really derive from those that Phil put in place. And the most important step that Phil took as director was recruiting a group of existing MIT faculty members to be the founding faculty members of the McGovern Institute. And they have continued to be the core and the guiding influence of the McGovern Institute over the years. Anne, Bob, Nancy, Emilio, Martha, Tommy have just been a tremendous source of inspiration over the years. And I think one of the things that Phil pointed out was the breadth of research represented in the core faculty members. The breadth spanned from cellular, molecular neuroscience to systems, to cognitive neuroscience, computational neuroscience. And I have to say that this idea that Phil had was pretty controversial at the time. I think there was a lot of thinking that the McGovern Institute should pick a more narrow range of neuroscience and just keep focused on that. And I think that Phil's insight was that the neuroscience of the future was going to depend on us understanding the brain and mind at multiple levels. And that has guided the further development of our faculty. They span all these levels. And again, although I think it was controversial at the time, these days when I hear of new neuroscience institutes being established and expanded and so on, they're all following this sort of model. The multi-level with people, some people sits at the intersections of levels, but certainly everyone is communicating across levels, and that is what is going to enable us to go from the mind and brain down to an understanding that goes down to the level of cells and the molecules and genes in cells. So I'm just going to review really a few of the highlights of work over the years, and I'm leaving out a tremendous amount of stuff, I acknowledge at the outset, but I just want to really give you a sense of the kinds of things that we've been doing. And I'm going to adopt a theme that I'm actually trying out on you. And the theme is that we can describe what is taking place in neuroscience is what I would call precision neuroscience. And I'll be explaining what I mean by that. And that this development of precision neuroscience is leading us to what I'll argue is going to be precision applications of neuroscience. And those include in health, education, and technology. And I'm going to give you some examples of that as well. Now, I'll take as one example of what I say precision neuroscience at the level of spatial localization. It's taken from the work of one of our junior faculty members, Mark Harnett, who goes in and records from inside individual neurons, and not just individual neurons, individual pieces of neurons, and is able to uncover the nature of computations that take place in cells. Now, I would say the standard thinking in the field has been for many years that the important computations of the brain take place at the level of neural circuits, lots of cells interacting with each other. But one of the things I think that's really exciting that Mark is discovering is that many of these computations are actually taking place within a neuron. They actually carry out very complex computations based on the processing within all their dendrites, all those the pieces of the cells that you see there. Another thing that he's done is by comparing neurons across species. Most of his work is in mice, but he's now going into human neurons. He's taking brain tissue from patients who have undergone brain surgery over at MGH, and he rushes this over here to the building and can record from human neurons, and is finding properties of cells which may give human neurons an even greater level of complexity at the single neuron level, and his work is beginning to explain that to us. Now, another example of what I would argue is the increased spatial precision of neuroscience is from the work of Ed Boyden and his work in expansion microscopy, where he's able to take a piece of brain tissue, and we hope someday maybe whole brains, and expand them up so that when they are examined microscopically, we really can see large volumes of the brain imaged at unprecedented detail at the level of all the synapses and processes of cells. And now as Ed is taking us even a step further and is able to now spatially localize a lot of even the molecules and gene transcription factors and so on within all these cells and their cell parts. And I think Ed's work is going to truly give us the Google map of the brain down at the level of the individual street corners and signs and so on. And this is really going to be critical for understanding how all this works. I'll take another example from Ed's work that comes from our increased temporal resolution in our ability to study the brain, and that's, of course, Ed's work in optogenetics, which has absolutely revolutionized neuroscience. He's one of the co-founders of optogenetics. This ability to genetically alter cells and then stimulate cells with light, controlling them with millisecond precision, which is giving us this ability to understand the operation of neural circuits. And even now, we're beginning to see the development of clinical trials making use of optogenetics in this way. Now I'm going to jump levels here and talk about our spatial precision and understanding the brain really at a global level and in the level of the human brain. The examples from the work of Nancy Canwisher, who you'll be hearing about from later today, who is showing us in the area of perception how very complex functions, such as the recognition of faces and colors and scenes and so on, are taking place in functionally specific circuits that are also functionally localized. She's beginning to understand how these circuits interact with other circuits, how they develop in development in both animals and people. I think that this is giving us a sense of the general operating principle of the brain, which Nancy describes as the Swiss Army model of the brain, with specialized circuits for specific functions, that is in contrast to, I think, sort of the conventional view that the brain is like a Pentium processor and pretty much all the parts are doing the same things, just running different programs. And really, it's what Nancy's work is showing us is that that's really not the case. Taking us up even to another higher level of abstraction is the work of Rebecca Sachs, who's looking at the functional localization of even more complex cognitive functions in the human brain, and you'll be hearing from her later today. She's looking at the functional localizations of processes like the theory of how other people are thinking, for example, and how this relates to social perception and social cognition. And now she's even taking this to the development in children, how these concepts that are so important to what it means to be a human being, how they develop in children, what's necessary for them to develop in children to become adults. So, as I mentioned, I see this progress in precision neuroscience is ultimately leading us to precision applications in education, health, and technology. And to give you an idea of what this means at the level of education is the work of John Gabrielli, who is using brain imaging in humans to understand childhood development and childhood disorders, such as dyslexia, for example. Now, it turns out that dyslexia is a very heterogeneous disorder. There's many different types of dyslexia, and John is using brain imaging to sort of dissect out what's going on in different subgroups of kids with dyslexia, and he's actually even extending this beyond dyslexia into trying to understand the brain systems involved in, say, distinguishing between good readers and poor readers, for example. And he's even identifying that some of these systems can be identified in children even before they read, based on the connectivity of these areas. And what I think we're going to be looking forward to in the future is the early markers that predict which kids will have learning disabilities of different types, and that we will then be designing interventions to address those before the kids even begin their formal instruction. And I think that this is going to be true in many aspects of early education, is identifying which kids will benefit the most from different types of educational experiences. Now, we're not going to be giving brain scans to all of our children as much as they need them, but once we begin to understand the neural systems, it also allows us to these are like biomarkers, is behavioral markers that can be done much easier and cheaply in children and get them on sort of the right educational path. I want to bring up another example of what I see as leading to precision education, is a collaboration with John Gabrielli and some of our Chinese colleagues here from Beijing Normal University, which was done in a Beijing kindergarten. And what we did in that study was to test the effects of early music education in comparison to extra reading instruction in these kindergarten kids, and use EEG measures to probe how these kids were processing information. And what we found was, maybe not so surprisingly, was that the kids who had the piano education became better at distinguishing between tones. Okay, that sort of makes sense. But also they became better at distinguishing between some of the sounds in words, like for example, distinguishing consonants, which is really the foundation of language development in children. And we know that in certain types of dyslexia, this is specifically impaired. And again, what our plan would be is to identify which children might benefit the most from this kind of early music education and target them so that then, this gives them an advantage in their early education, and those studies will be getting underway. Now, as another example of what I take as the spatially precise applications in health is the work of Anne Graybill, who you'll be hearing about today. Anne Graybill has been identifying with neural recordings and anatomical methods the specific neural circuits that are important in, for example, in anxiety, in affectives, and valence, positive and negative valence, rewards and punishments, and so on. And now, having identified these circuits, she can then target them. And in collaboration with our colleagues Bob Langer and Michael Sima, she is developing these incredibly small devices that can both record neural activity, record even neurotransmitters like dopamine being released in cells, and then apply minute amounts of drugs to just those specific circuits. And they are now, in this collaboration, developing this device for use in Parkinson's disease, for example. And so what we're looking forward to is applications targeting just those cells that are involved in the disorders and not necessarily bathing the entire bodies in pharmaceuticals. Another example of precise applications, of course, is in the field of genetics. And throughout the field of genetics, what we're learning is that medical categories, that the categories like that we use, like bipolar disorder, schizophrenia, autism, depression, and so on, these are broad labels, but they actually encompass many different biological disorders that have different genetic signatures. In autism, for example, we know that there's a variety of genetic mutations that contribute to vulnerability to autism, and some of them have shared biology and some of them may not. And that means that the therapies of the future are going to have to address the specific disorder that someone has. And Feng Zhang, who's developed the revolutionary new tool known as CRISPR, is developed this technology to develop those treatments for specific genetically defined disorders, in both as a research tool, but also we're now seeing increasingly even in clinical trials using CRISPR-related technologies that are going after RNA editing in adults, for example. And I think over the next few years, we'll be seeing more and more clinical applications of this technology. In a related way, Feng is using these CRISPR-related methods to detect disease-causing agents and detect vulnerabilities to disease. So, for example, he can use what is now is very inexpensive, easy-to-apply technology that can be used out in the field. It can be used in developing countries to detect infectious agents like Ebola, Zika, for example, and as well as disease-causing mutations and cancer and mutations that lead to vulnerabilities. And I think this kind of approach is going to have a big impact on healthcare in the upcoming years as well. Guoping Feng, who you'll be hearing from later, is taking these CRISPR-based technologies and using them to develop better genetic models for disease. Now, of course, the work in mice has always been the foundation of biology and neuroscience, and will certainly remain so. But one of the disappointments of the work in mice has been that many treatments developed in mice have not translated into applications that can be used in people. And so Guoping is developing advanced primate models of these brain disorders. And we have, as our first major project that was done in collaboration with our colleagues here from SCIAT, the Shenzhen Institute of Advanced Technology, we have a paper at review under a major journal reporting our first group of monkeys with an autism-related mutation, SHANK3. And these monkeys show a variety of behavioral abnormalities that resemble a lot what is seen in this particular form of autism. They have sleep disturbances. They have MRI signatures of brain connectivity differences that's giving us new insight. And over the next few years, what we anticipate are these genetic models being developed both in China and here in MIT will be used for both developing and testing new therapeutics, whether it's drugs or whether it's genetic technologies. I guess I used the wrong slide. Yeah. It's a problem with seeing from over here. I was using the wrong slide for talking about disease detection technology, so I've already given you that. So I want to turn to, lastly, another application of what I've talked about as the precise applications of neuroscience, and that is in the development of technology, particularly development of artificial intelligence systems and these neural models, which then give us insight about how the brain works. And one of the leaders in this field has been Tommy Poggio. For example, he has developed artificial systems for visual recognition, and some of his students have applied this general approach to developing collision avoidance systems and self-driving cars and so on. And Jim DiCarlo was taking this work even a step further in developing these artificial models which have properties that resemble those of the neurons that he records in the primate brain. So, for example, he will take these models, train them on massive databases of images, like you see the chairs here, and they learn to recognize these. And then he will find the models who have properties that resemble those of the neurons that he studies in primates. But of course, the question that is often raised by this kind of work is, well, but what's the biological relevance of these models? I mean, how do we know that this artificially developed model actually resembles how the brain actually does these same computations? So in recent works, he's done a really fantastic test of this idea in that he has taken these models that have developed these neural properties that seem to resemble those that he finds in his recordings. And he said, okay, so if this is a really biologically relevant model, we should be able to use the model to predict what will happen if we show the monkey and therefore these neurons images that they've never seen before. And we will predict the neuron's response from the model. And so he'll take this, for example, the model's prediction of the neural activity caused by the cat, and he can look at this activity across the population. That bar graph up top is just an activity measure recorded across a group of cells. And one of the things he's asked is, well, let's say that we want to make one neuron in this population give a more selective response. And so it'll respond more and its neighbors will respond less. Can we use the model to predict what would that image be? And it turns out that this image down here on the right, what seems like a bit of nonsense, but the network came up with that, and it predicted that if you were to show the monkey that pattern, it would give you a more precise activation pattern in the population, and indeed, that's what he finds. The actual population he records resembles what the model predicts, suggesting that we're on the right track. Now, one of the other, I think major insights that Pat had was that these problems that we've been talking about, understanding the brain and helping people with brain disorders and so on, is going to require an international effort. It's not all going to be done here. And so Pat and Lori, with the help of Hugo Sheng, decided that they would establish brain institutes in China who they felt were going to be making a major contribution to neuroscience in the future. And they established these 3 McGovern institutes in Beijing, who are all represented here today, and we'll be hearing more from them. And we've now developed this cooperative research center in Shenzhen that's developing the genetically altered primates, for example. And we've seen now this developing of this international network for neuroscience that I think is already beginning to pay off in neuroscience discoveries that we'll be hearing today. And I think one of the things that's really encouraging about the development of the neuroscience of the Chinese institutes is that they're following this same multidisciplinary concept that we're applying here. In other words, they're studying the mind and brain across multiple levels of scale. Many people are sitting at the intersections of different techniques, molecular and systems and so on, and they're establishing institutes where people can then talk across all these levels. And I think we have become confident that these kinds of interactions across people will enable us to understand the interactions in the brain. And now, I would say, Pat, if you've been listening, I hope that you accept this progress that we've been making from what you got started. You and Lori started here at the McGovern Institute, and that maybe you would give us some encouragement. Glenn Rifkin described the famous memos that Pat gave at IDG, the good news memo, telling people how wonderful things were going and suggesting how they might make it even more wonderful. And I can imagine if Pat were here now, we would be getting the equivalent and giving us encouragement in the future as well. So thanks, Pat. And I will turn it back over to Sabi.